Welcome to the second half of Chapter 39's podcast. So right now I want to start off with the plant hormones that you guys got to know some about. So the first one's going to be auxin. You might see it called IAA. It's just an abbreviation for, I think it's Ethylene acid or something, but that's just its fancy term. But auxin, and so this one was discovered fairly early, and this one's primarily associated with growth. You'll see the first three we talk about are really focused on growth. And so you'll find it in tissues that are growing. Notice that meristems are just regions where cell division, where growth is occurring. Apical just means it's vertical growth, so the plant's growing up. There's also going to be auxiliary or lateral growth they'll talk about, or auxiliary buds, auxiliary meristematic tissues, and those are going to focus on growing out. So plants typically grow apically initially. If you start to trim them, like a lot of people cut back a plant, they'll start to grow out because they have to use those buds that are further down. They can't use that topmost bud. And so that's why a lot of gardeners will trim their stuff back to make it get bushier, not just really tall and skinny. And so young leaves, etc. Now these guys in stems are going to elongate the cells. So that doesn't mean cell division. What it means is the cell initially, let's say it's kind of more squarish, they will cause it to take on water, and ultimately they likely produce some acid and kind of wear away the wall, and then they allow things to just expand. And so eventually this cell can end up being a lot longer, up to about 10 times longer than it was originally. And so because of that, we do have growth. But the growth is from elongation, not cell division. And this is what happens in stems that leads to the tropisms. So things like phototropisms, gravitropisms, those are going to be tied to this idea of stem elongation typically. Although obviously with gravitropism, there's a bit of that that's going to be in the roots. In roots, it makes them grow and it makes them spread out, branch. And we've already said this is tied to apical dominance, so this is trying to make the plant grow up. And so initially when you see a plant first sprouting in that, you'll see it has that kind of mad dash for the light typically. And many plants continue to just grow up and up. You know, you've got like bamboo and such, but they're not bushy. They're very much just going up. If you leave a rose plant be, you'll see a rose plant will keep going up and up typically. They won't be very full. And so that's going to be done by auxin. Now, if you want to make things bushier, you can focus on cytokinins, because these guys, they will do cell division. That's a big thing for them. Still growth. You still find them in our tissues that are growing, etc. They also do focus on germination. They delay aging, which is kind of a cool thing for them. They can kind of keep things going and young. But they'll play a role in apical dominance. And how this works is if I have a lot of auxin and not a lot of cytokinins, they tend to have the plant grow up. If we tend to cut that top tissue so there's lower amounts of auxin, we have more amounts of cytokinins, it triggers these auxiliary, these extra, these lower meristematic tissues called buds. They will start to then grow, and so that's why we said a lot of people will go through and cut the top part of these plants to force that to happen, because they'd rather have a nice big, rose plant that's wide and short rather than just having one that goes 10 feet up in the air but is spindly. And so it's very common that people will use that because of it. So cytokinin in general growth, but it plays a role in the seed starting development, germination. It plays a role in general in delaying aging to kind of keep things from getting old as a plant. Some plants can exist for ridiculously long periods of time. So there's nothing saying a plant has to age like us and ultimately die after a year or a decade or whatever. And then apical dominance with auxin. Now, gibberellins are more of a general growth hormone, I think of it, where plants that have a lot of gibberellins tend to get to be more giants, and plants that have very little is how we get a lot of our dwarf varieties of plants, because we just either inhibit or in some other way prevent them from having gibberellins, whereas if you want, you can spray gibberellins on plants, and you can get taller plants. And so these will be associated, once again, stem elongation, general growth. You see it in these growing tissues, meristems, embryos. But they will also be part of flowering and fruiting. Flowering you might also see called bolting. So if you see the word bolting, that's usually just referring to flowering, so don't freak out, just the same thing. So each of these have kind of their own specific things that are kind of unique to them. And so if you see me talk about flowering or fruiting, you can usually figure out, all right, that one's going to be gibberellins. If you see me talk about general growth, it's a decent chance it's gibberellins, especially if we're like spraying it on a plant. You'll see that there are things where you can spray or put gibberellin like in the soil so the plant can use it, and it will cause them to grow more. I don't usually hear about that with things like cytokinins or auxin. Okay, abscisic acid, this one's different. This is not the same. Abscisic acid is largely about dormancy, so it's about keeping the plant inactive, such as in seeds, such as in fruit that's ripening, where you're trying to kind of say, all right, chill out, let's just relax. This is not saying grow, grow, grow. It's saying wait. It also will close the stomata if there's water stress. So if we're looking at that whole transpiration, photosynthesis compromise, where you're trying to grow but then you lose water, if you start to lack water severely, you'll produce abscisic acid and it'll just close things up. And you'll say, all right, we don't want to die, so we'll shut our stomata down and just do without as much photosynthesis. So inhibit growth, that's tied to dormancy, but it's basically just saying, wait, is kind of what this one's whole thing is. Now, with ethylene, this one's major claim to fame is it's a gas. And so this one's produced by ripening fruit, and this is where the expression, one bad fruit can ruin the barrel, because as a fruit ripens, it's producing this ethylene gas that can then diffuse to the other fruit, which can then trigger their ripening. So this is why some people will go through and spray a wax coating on the fruit to try to prevent it from going bad, because it tries to seal it up, so you don't have to worry as much about that ethylene affecting it if something goes bad. It also plays a role in aging and leaf abscission, which is where in the fall you see the leaves start to kind of wear away as they age, things are getting rough. It's no longer beneficial to start cranking out new leaves to replace these older leaves that are in some cases damaged. In some cases they're just not worth it. So what we'll do is we'll wall it off, and so we're just going to take this leaf where it comes off, and we're just going to build a little miniature skin layer, which is what the abscission part is, which allows this leaf then to fall away. So it's kind of like in your arm if you kind of built a whole layer of skin and everything here, so the rest of your arm just falls away because we've isolated it. That's what goes on in leaf abscission. So aging, ripening, abscission, and if it's a gas, that's definitely ethylene. Now, plants can also figure out what time of day it is, so to speak, or at least what part of the day it is, and they can detect seasons. The circadian rhythm is going to be this kind of daily pattern of light and dark. It's going to be this fact of I know when a day is. So circadian rhythm is more daily, not so much seasonally. And photoperiodism is when we use this data about individual days, which we can use the amount of light and dark in an individual day to figure out the season. Because you've got summer and you've got winter where you have a fairly consistent, even amount of light. And then you've got, let me say that one more time here without being an idiot. So you've got spring and you've got fall where there's a fairly even amount of light, and you've got summer where there's a lot, and you've got winter where there's a little. And so they can use these different amounts of light per day to figure out what time of the year it is. And they can base decisions on that, such as flowering. So a short day plant is really a long night plant, because plants really focus on the night part of things. And so if the night is longer than a critical length, there'll always be this thing called the critical length or period. We'll say period just to be fancy. If it's longer than that, then that means you'd start flowering. So you might say if the night gets to be longer than 10 hours, I'm going to flower. That would be a short day plant. I know it says short day, just think the reverse. It's actually a long night. And then the long day plant is actually a short night plant. So it might say if that you have something that's the night is shorter than 14 hours, then I'm going to go through and I'm going to flower. Could be shorter than 10, doesn't really matter, but it's shorter than some critical amount of time. That's when it starts to flower. And so based upon the exact length they have and what they're going for, this can allow you to flower in the spring, the summer, the fall, whenever it's advantageous. And then day neutral plants, they don't care. They're going to base their decision on something else like nutrition. It's not going to be based upon the light. So day neutral plants will flower when they're good and ready. Now phytochromes are the guys that detect kind of whether it's day or night. And they do this because they can switch between two forms, PR and PFR. Now PR is going to be formed when red light's present, which would be during the day. Whereas PFR is formed when this infrared's present, which would be during the night. So at nighttime, the PR gets converted to PFR. During the daytime, PFR gets converted to PR. And so plants are going to focus on measuring the amount of PFR mostly because they tend to focus more on how long is that night. And this is useful because if you have a neighbor that you really hate and you find out that he's got a short day plant, which needs a long night, longer than a specific period, if you really wanted to, you could go through and shine lights onto his plants at some point during the night, And what would happen is that plant would no longer see one long stretch of night. If you shine lights in there, it would see, oh, there was a brief period of day. And so now we've got two short nights, neither of which is long enough to make it flower. So if you were really into this, and I don't know what would have to be wrong with you to really hate a person enough to do this, you could make it where particularly their short day plants would never flower. The second part here is there are other photoreceptors, there are other things detecting light. It's not just about red light. I know the whole thing here with flowering and that is tied to red light, but there are other receptors such as these blue receptors that determine kind of when the day has started. So this is how the plants say, oh, it's daytime. You know, we can do photosynthesis. Let's open up our stomata. It also will be determined for phototropism. So if I shine a yellow light on a plant at an angle, it won't grow towards it. If I shine a red light at an angle, it won't grow towards it. But if I shine a blue light or a white light that contains blue, it will. So it's specifically just one chunk, one set of wavelengths that will determine whether or not it reacts. Don't assume plants can detect all lights for all things. Most things that are detecting light for a plant, most of these proteins that are doing it, they're going to be still relatively specific, just like our photosynthetic pigments, about exactly what types of light that they're able to interact with. So that's no different. This just shows you a picture. As I angle the light, you'll see the auxin that's being produced at this apical meristem at the tip is no longer being evenly distributed. It's now being moved to the shady side. And as it goes to the shady side, that shady side will now elongate more. And so because it's elongating more, that's going to cause the plant then to have to bend to the other side. Because the shady side's getting bigger, it's got to push it to the non-shady side. Just like we said before, one leg gets bigger, you get pushed to the other side. And so now at this point, we have the plant is aligned better. The benefit of this is you get more direct sunlight exposure, you can do more photosynthesis. So imagine if someone was like trying to throw food in your mouth and that's how you ate, that's the only way. And I have my head turned at an angle to how they're throwing. It's going to be harder for them to get food into my mouth. So I'm going to eat less. If I could just turn my head towards them, now we've got more of a direct pathway, I'd be able to eat more, and that's probably going to be a good thing in terms of my survival. So that's really what plants are trying to do here, is just benefit their chance at living. Now, general plant defenses, the simplest things are physical. So this is going to be like an epidermis, or their skin, if you will, as well as thorns. And so this will keep out basic pathogens. This will at least deter many herbivores that would like to graze on them and get their nutrition. They also have chemicals. Phytoalexins are kind of more general chemicals. You can think of these a little bit more like antibiotics type substances. We have some of these antimicrobial substances that we produce. They're not phytoalexins, but similar, that we secrete in our skin, etc. And then the PR proteins are more specific. I guess you would say these are a bit more like something like our antibodies. They're more targeted to specific pathogens or groups of pathogens versus being more broad. And then the other way they can do it that's really cool is many of them will enlist animals. And so you've got some of them enlist ants. So if they're being damaged, they release chemicals that the ants detect, and then they can run over and kill whatever it is or kick it out. Some of them will even patrol the area and kill any plants that start to grow. You've got some of these that can detect things like caterpillars damaging them and release chemicals that attract parasitic wasps that attack the caterpillars. So that way the plant wins, the wasp wins, caterpillar loses. So there's lots of ways that they can interact, not just even with themselves, but with other organisms to allow them to be able to protect themselves against stuff that would eat them either from the inside out or literally just grab a chunk of them and munch it like a lot of herbivores. Now, specific plant defenses, plants can oftentimes detect specific products that are produced by pathogens. And so this is called gene for gene recognition, where I can detect a protein or something else that's on a specific pathogen so I can activate the correct PR proteins or whatever chemical pathway I need so I could try to go after them. You can also, if you get this path triggered, such as the systemic acquired resistance, but if you trigger something, salicylic acid, this is just a chemical that if a plant is under attack, if it's being damaged, it commonly releases this. And so this triggers the plant to kind of shift into this mode of producing things to protect itself. It can also trigger the release of volatile chemicals, or they can be released by chewing. These are just ones that go off into the air. So these can then enlist other animals. They can warn other plants nearby. And they can also cause a hypersensitive response, which is where a plant says, all right, this branch, which is part of my tree, this is a horrible looking tree, but this branch is the one that's infected. So they can actually wall it off and just say, you know what, screw that branch, it can fall off or sit there and rot. I'm going to build this wall so the rest of it's protected. And so that's one of the things that can be triggered when they detect this damage, when they detect the issue. So it's part of their response that they can have to resist any invasion by a pathogen, be it fungus, bacteria, whatever. That's all part of their repertoire to try to keep themselves protected. That's it for this section. We're done with 39, and we're, at least for this purpose, done with plants till we get to ecology more. Hope you guys enjoyed it.